Hello.
Hi, how are you?
I'm pretty good. Yeah.
Yeah.
I haven't started into any heavy duty research so far. Yes, I haven't managed to start my paper. Well, I started it, but all I did was a summary of my thesis in 2 paragraphs. And I'm supposed to be writing an overview of modeling epithelial tissue, like who's doing what type of modeling. So I'm supposed to do a literature overview. And so far, my attempts at getting papers has been 0. So so I've asked Dr. Sharif, who I was working with, if he would mind if I could work through his lab to get papers from the university.
Okay. Well,
the only other thing is to ask Dick, I guess, to get the papers, but that's not quite fair.
Yeah.
Yeah. So were you on last week or the week before? Or?
No, I was off the 2 weeks ago, and then last week I did an update, but it was like no one showed up, so I did a kind of a YouTube update on things.
Oh, okay. So I could look for that. And I'm also supposed to be finding some more salamanders so that I can do some experiments on eggs. I'm lining it up to do optical coherence tomography on salamander eggs.
Oh, okay. That sounds good.
Yeah. Well, it should be a worthwhile experiment. And like I told Dr. Sharif that it'll be a paper, like get more than one image of the salamander eggs or more than one egg and several images of the eggs. And it'll be worth a publication because nobody's done this.
Yeah.
They actually did one of xenopus eggs, and I can look at how they did that and kind of match it, but I'm going to also look at the density of the salamander eggs at the same time as looking at their blastoceles. You can't really see the cells with the optical coherence tomography, like the definition of where the cell membrane is.
Yeah.
But you can certainly see the blastocele. So you can see the blastocele develop. Mm-hmm. So I thought that was worthwhile. Get a baseline. This is blastocele development in axolotl salamander. I mean, it's definitely worth a publication.
Yeah. Sounds good. Yeah.
And then you can look at other things that the eggs are doing, like perhaps some cell cramps across the surface, maybe distorting things a little. Anyway, yeah. I know for sure they have cramps across their surface and they develop. Mm-hmm.
Well, that's good. Yeah, good luck with that.
Yeah, I don't know how many I can do at once in a batch. I was going to say, I'll try 10 and see how difficult that is.
Yeah.
Because I have to take at least three scans across, like 3 sagittal slices, in order to kind of project the shape and size of the blastocele. Mm-hmm. And according to metrology, I should be taking 5 sagittal slices. That's the industry standard. Anyways, that's in one go. Like that's right. Yeah.
So I have an interesting paper today on tensegrity, and it just got published in Biosystems. So we'll have to go over that.
I found a very nice paper about shape and the shape of a developing embryo and the forces in it.
Oh, really?
Yeah, so I'll find it and I'll put it in the chat.
All right, that sounds good. Yeah, this is it.
Okay.
So this is studying gastrulation by invagination, the bending of a cell sheet by mechanical cell properties using 3D deformable cell-based simulations. So this is zoom in. So they're wanting to study the bending of a cell sheet in vivo, so with in vivo imaging. And so they basically have to separate out something like invagination in the action of the cells from other cellular processes and properties that interact. And so then we can do computer simulations. 2D computer simulations are unsuitable because they don't really take into account three-dimensional properties of the cells. We've talked about a lot of that, where you have the shape of, like, not only as sort of the top of the cell, but the sides of the cell, and how they twist into different shapes, and of course cell number.
Okay, now it's making a noise.
Therefore, we developed a 3D cell-based model that is able to simulate cells as separate deformable entities with a conserved cell volume. So they're fixing cell volume. They're, with respect to deformation, and they're able to have model 3D deformation of the cell. So like twisting and bending, but still having the cell body intact. And then they're able to form a blastula by adhering cells together into a sphere. The simulation results show that changing individual mechanical properties like cell stiffness, cell adhesion, and the apical constriction factor had a direct effect on the cell's behavior and future shape. So they could predict the shape changes, and they could affect or predict the behaviors more generally. These properties influence the ability of a cell sheet to bend and eventually change the global shape of the embryo. So all the cells in the simulation were able to have this collective effect of shape change on the embryo. The observed shape transformations, this transitions the endodermal region. It goes through a number of different changes during the inward bending of the cell sheet in the simulation. This can give insights into the mechanisms involved and the timing of events in actual biological systems. Changing geometrical properties, and they give a number of examples here, which is not possible in 2D models, showed that the inwards bending is more dependent on the number of cells involved than on the shape of the endodermal region, and thus that the invagination process is very robust to irregularities. So this means that, it isn't, the invagination process is usually regular across embryos and unfolds in a predictable way, and it's because it's robust to these irregularities. When quantitatively comparing our simulation results to biological data from the literature, we saw that our simulations did not exactly reproduce the shapes observed in nature. So, that's probably normal, but it's capturing a lot of aspects of that. So there are probably additional mechanisms at play, and that accounts for the difference in the results from being exactly like nature. So this is a blastula here, where we have the blastopore, and then we have the endoderm here, the ectoderm, and the blastomeres out here. So they're kind of showing the blastula as it occurs in nature, and then the gastrula, where there's this inward folding, this invagination at the oral animal pole. So the oral pore is formed at that pole, and then you get the same kind of structure, but you get this folding inward into this shape here. which is the gastral. So that's what they're trying to mimic, those kind of shape changes. And then this is shown kind of in microscopy. So this is just showing, this is in Nematostella vectensis. And so this is just showing this, these changes in that organism. Then this is their model. So zoom into the model here. So basically they have, let's see, go up to A here. It's kind of like a poster. They have a number of properties of the simulated cell. The cell has a cortex, which is the outside, which consists of edges, interconnected vertices, and are deformable. So you have basically this skeleton here where you have a number of vertices with edges. They're connected by edges. And then you can deform this shape. So you can deform this triangle. You can move one vertex inward. You can move it outward. And it shifts these edges, their length and their orientation. And then that's relevant in forming this deformable mesh. So each one of these triangles forms an element in this deformable mesh. And then when the mesh deforms, say when something pushes on the entire surface of the mesh, all of these triangles deform in a coordinated manner. So if I put a weight here, if I introduce some force right into this area of the triangulated mesh, it would push everything else down and deform all of the vertices and edges with respect to position and especially the length of the edge. So you can see that that's kind of how they're doing this. They're putting then this triangular mesh on the surface of a sphere, which is the embryo. And so you're able to measure, or you're able to simulate the cells, you're able to simulate the surface, and you're not including a lot of interactions, that are sort of there that you can't directly measure. You can just look at how the cells sort of respond collectively to mechanical properties of the embryo environment, and then map that onto the surface of an embryo. So when the surface phenomena happens, that can be simulated in silico.
Oh, okay. Well, this is again. They're probably using continuum mechanics.
Mm-hmm. I think
and so. They're using triangles, which are the most stable structure, the most stable structure.
Yeah.
So it's probably will give different answers from real life. Mm-hmm. It's, I guess, approaching it, but their cells are hexagon-ish.
Right.
Meaning that they're not always hexagons.
Yeah.
Anyways, they tile, like right. Triangles certainly tile a surface, hexagons tile a surface, five-sided objects plus a seven-sided object tile a surface. Mm-hmm. So, anyways.
Yeah, and there are no gaps, which might be true and it might not be true in biology. Because we can have gaps, I guess.
In embryology, there's definitely tissues that have gaps. Like they're not solidly tiling something.
Yeah, and I mean at any one time too. So like you could have gaps that are sort of itinerant, and
it changes. I think I need to look that up, but it was in one of the APS meetings one time, and I think the tail is disassociated. Like it tends to be.
Yeah.
Maybe that's why it elongates. I don't know. Anyways, it's interesting.
Yeah.
And interesting to study that. Yeah.
Yeah. So let's go over a little bit more on this figure. So yeah, there's this resting length, and then you can pull and you can push on the edges. So the pushing is, or I guess the pulling is outward and the pushing is inward. So it's just like, once you change the length of these edges by these sorts of forces, then you have, and that translates into pulling and pushing respectively. Okay, so then they show this kind of like the direction of the forces. So they actually have sort of this force orientation map where we have changes in the sort of the forces on the surface of the sphere, and then changes in the sphere itself. So the cells, the entire embryo can grow, the cells can grow, things can move inward or outward depending on the mechanical um, environment. And then in this C, they show the adhesion region, which is where basically they're showing this transition from blastula to this form here, where you have the gastrula, this pore here that's coming inward. The embryo is kind of changing its shape at the top, and it's going to form this pore, which then has some internal structure, which isn't what they're really modeling. What they're modeling is the outside of the embryo and this shape change. So, you want to be able to understand that part. But this adhesion region changes as the shape changes, and that's the important part. So there's this effective constriction on the shape of the adhesion region. It changes kind of from this example here in the sphere to this example in the partially constricted embryo. And then we have vertex restorative forces. So we have these three cells that are kind of linked together. They're in contact. You can see how they model the cell body as a series of nodes and edges. Those deform, and you can see where there's an adhesion point where the nodes of cell A and cell B are sort of overlapping, and it sort of, each part of the edge of each cell is constricted in terms of shape. So if they bump into one another, they push into one another and form kind of like this. It's almost like a soap bubble where when they bounce together, they kind of form a flat edge or they form a flat boundary. And that's what you're seeing here with adhesion. And then with cell B and cell C, you see collision. So you actually, in the simulation, you see a collision where there's no adhesion, where the nodes and edges for cell B and cell C overlap. And so then that's a violation, of course. So they have to have this boundary violation resolution, which is where repulsive forces are proportional to the depth of vertex penetration. So you have this repulsive force that acts when there's some collision of some depth. So that's basically how they manage that in the simulation. So they basically manage this as an additive model where they have the total forces are each element plus the volume plus repulsion plus adhesion. So all these components go into the sum total of forces. So there's the force of adhesion, there's the force of this repulsion as a result of collision, there's each element, and then the volume itself. The displacement is found by using Newton's second law of motion together with the Verlet algorithm as the numerical solver, and then this is the equation here. So yeah, and then the pulling vertices together, you have this force of adhesion, which is, they have the physics here where, if you have this certain distance, you have adhesion, and that pulls these edges together so that you have these cells that don't have gaps in between them. But it's interesting how they have to deal with these edge cases like collision, and they resolve that by doing some sort of repulsion, and that's sort of correcting when you don't get adhesion the way it's supposed to happen. Now we don't actually, this may happen in biology somewhat. Obviously they can't overlap, but you can have cases where two cells will collide and bounce off of one another and then have to sort of form an adhesion, and the adhesive forces aren't strong enough. So that's kind of what they're getting at. They're trying to resolve cases where they can't get adhesion using this method. And then they're multicellular simulations. So you have, these are the cell bodies. You have this, the first case here, 128 simulated cells. You have endoderm, the cells that are in salmon on the left side of this sphere. And then you have ectoderm and beige, which are the ones over on the right side of the sphere. So this is simulating the blastula, where you have the shape and the direction and everything. The cells are here. They actually have some gaps in here, as you can see. And they're going to form then the second 128 simulated cell simulation, where the cells are adhered to each other in these hexagons. So you see in the spherical cells coming together to form this embryo, there are these gaps. And what they're trying to do is resolve the gaps. So if we go back to where the cells are adhering, they haven't adhered yet. They're just kind of in this form, and so they have to adhere for this, to model the physics properly. So here's where they're all adhered and all resolved. So now you have these hexagons, and there are no gaps within the body of the embryo. So it's forming this multicellular structure that has like kind of a constant contact between everything. And then this is the third example. This is an internal view of this structure. So if we take the second structure and look at like a cross section of it, we can see adhesion zones, and these are colored in light blue. So they pull the cells together slightly apart, and they want to show the cell-cell adhesion. Cell-cell adhesion are in these black lines. And so that's the idea, that there are these, there's a cell-cell adhesion process. They don't model all of the molecular components here, of course, but they're kind of modeling these adhesions as physical phenomena, and then just, kind of showing where that would be. Okay.
In an axolotl egg, you actually get, you can see them turn from slightly rounded to flattened cells on the surface. Yeah. So, yeah, which is the middle part there.
Yeah. So that was figure 3. And then they kind of go through constriction, which is this figure here, constriction modes. So basically they simulate this and show all the different modes of constriction. They show cell stiffness and constriction factor. So they vary the cell stiffness to see if that, how that plays a role.
Yeah, I'd like to know what software they're using.
I'm not sure. Let's see if we can find it. Methods.
I haven't read this.
Yeah, it'd be good to see it. Okay, so they're using a lot of math, I see. Really they're giving a lot of detail, but they're not giving any information about the environment that
Did they make their own?
They may have. I'm not sure, though.
Yeah, well, a lot of people do.
Yeah. Which probably isn't good, because you need to have a standard.
Yes, it would be nice.
Yeah.
Nice to have some sort of standard.
Okay, yeah. So I don't see any information on the environment other than because they said our vertex model was specifically developed to simulate the bending of a cell sheet in a blastula. So they developed this, I guess, homebrewed manner.
Looks like it.
Yeah. But yeah, and then so they're able to do a lot of interesting things here. But again, this adhesion part was interesting because, both in a good and a bad way. Because on one hand, you model adhesions, but on the other hand, it's just kind of like these lines. It's kind of like, oh, there's an adhesion. What does it involve? Well, it's a process, but I don't, I'll just kind of draw a black line, or we'll model it as a single process. And
it's more complicated.
Yeah. All right, so I think that's good. Thank you, Susan, for showing us that paper.
Thank you, Dick.
Yeah. Oh, okay. Thank you, Dick. Okay. Why don't we go to the Biotensegrity paper, which I know you've been waiting for.
Would it be possible for you to send me that reference?
Yeah, I will.
In an email, because my computer's is limping along.
Yeah.
I'll try to get it resolved soon. I think might be it's some sort of blocker that's blocking the Microsoft from downloading.
Oh.
Might be my ERO. EERO?
Oh, I don't know.
I'm not sure. I have to get to the bottom of it.
Yeah. Okay. So this is the Biotensegrity paper. I'll send you this reference. This is Stephen Levin is on this. Susan Lowell de Solorzano is on this.
And Graham Scarr too.
Yeah, Graham Scarr.
He's into tensegrity and biology.
Yeah. This is Biotensegrity is the superstability hypothesis for biology. So this is a theoretical paper. This is in biosystems, of course, being that. So this is, let's go through the abstract. Biotensegrity models living systems in ways that were inconceivable in the past, but has taken some time to become widely accepted because it challenges generally accepted wisdom. So orthodox biomechanics is essentially based on mechanistic models from the 17th century and allowed oversimplified representations of anatomy and motion to persist to the present day. So there's a lot of, if you look at modern biomechanical models, it's based on Newtonian physics and kind of thinking about a biological system as a mechanical system, with physics, but then, they don't really have a lot of biological terms involved, nor are there emergent phenomena usually. Those aren't really discussed in a orthodox biomechanics model. So that's their point here. Living organisms, however, are hugely complex, intrinsically interdeterminate, and exist in states that are far from equilibrium. And although their simplification within the machine model has enabled great progress in the mapping of structure to function and benefited our healthcare system in remarkable ways, it has also obfuscated the foundational basis for stability, motion, and life itself. So then they're contrasting this sort of orthodox model with a biotensegrity model. And so biotensegrity is a conceptual framework that is founded on a fundamental set of self-organizing principles and includes all complexities of life at every hierarchical level, from viruses to vertebrates and molecules to the whole organism, with stability and motion controlled from within the structure itself and the homeostatic algorithm of superstability. So I guess they're arguing that there is this model that's a biomechanical model at heart, but it's also kind of more biologically plausible, as well as having these properties of complex systems and dynamical systems. So they talk about far from equilibrium, they talk about homeostasis, they talk about heterarchy, actually. So, those are all kind of buzzwords, but I don't know how they fit them together here. Here, anatomy is no longer reduced to a set of discrete parts, but becomes the physical representation of a hugely complex pattern of interacting force vectors, and which are themselves organized within a complex tensegrity configuration that enables each part of the system to adapt to its locally changing environment in the most energy efficient way. And that also remains stable throughout that process. So yeah, they're kind of hitting on tensegrity or biotensegrity as being based on a fundamental set of self-organizing principles. So they kind of bring this around to self-organization. They talk about tensegrity structures being the physical representation of invisible force vectors. Living organisms are intrinsically stable tensegrity frameworks. The closed chain kinematics of tensegrity replaces the contrived machine lever model. So, like, if you think about kinematic chains, they're closed kinematic chains that, you have forces that are propagated outward from a central. So, if you have forces that are generated at some point in at the sort of the origin of the kinematic chain, those forces propagate outward and a lot of times are not controlled in their output. So if you think about like ballistic movement of the arm, if you're throwing a baseball or something, you generate forces in your muscles and that force goes outward and then there's an output that is this ballistic movement. And so you can, they use this in robotic control where they try to model that as a kinematic chain and they try to control not just the origin point of the forces, but the end point as well at the end effector. But in a closed chain kinematic system, all of that is sort of self-contained in maybe like a sphere or some system where those forces are sort of managing each other, where they're balancing each other, and they're forming this self-organized structure that allows things to sort of persist in a certain state. And so this is kind of an interesting kind of way to think about this. And then, of course, the machine lever model is a classic model of mechanics. If you think about, go back to your simple machines that you learned about in elementary school, where you learned about different machines that do work. That one of them is the machine slash lever, where you have a lever that you pull and it pulls something up or down. So you can pull on a lever and it lifts something up. You can push on a lever, it pushes something down, and that it's basically transferring physical work. And so a lot of biomechanics is based on those kind of simple machines. What they're saying is that you can model things not just with simple machines, but with these kind of new structures or novel structures. And then finally, superstability as a general mathematical framework, can be done, and this can be biologically plausible and model biological transformations. And so they basically have this quote here: the principle of organization is the fundamental source of biological stability, which is an interesting quote because it basically suggests that you have to have some sort of stable structure for biological organization to occur. So if we think about multicellular systems, like we just saw in the last paper, all those cells kind of exist in, they could exist in a cluster and they could be kind of free moving in that cluster. They could just be kind of loosely associated, but you also have multicellular systems that are integrated, both structurally and functionally. What they're saying is that there's this functional interaction or interconnection. That's the key.
And modeling this with standard software causes problems because they think the building has broken and it's just the cells moving naturally. At least that's what I began to think of when I was modeling it, because I would say, no, it's not working. Yeah. And really all it did was do whatever tensegrities do when they change shape. So they're changing shape from one shape to another. And oops, the building's broken. Well, no, it isn't. It's just changing shape.
Yeah. So, yeah. Buildings don't usually change shape, but that's kind of the point, right?
Yes, that's the point.
All right.
Yes. So you'd have to reinvent the software.
Right, yeah.
In order to do that. That means software that works with something that's more movable than a building.
Right. So yeah, then they kind of talk about this idea of developing kind of a new way of thinking about this. And then the first part is we are not machines. So, there are a lot of mechanistic theories of, biomechanics where you're just thinking about the mechanics. And then, we have to think kind of beyond that. So, this is kind of getting into this. The problem is that living organisms are hugely complex, intrinsically indeterminate, and exist in states that are far from equilibrium. And although the simplification within the machine model has enabled great progress, it has also obfuscated this foundational basis for life. And then the ability to overlook the approximations, assumptions, and unknowns inherent within standard analytical methods is routinely accepted as part of the modeling process. Riley noting there are these citations here, or Bertrand Russell, I guess, is the source of this quote. Riley noting that all exact science is dominated by the idea of approximation. So that's kind of, we have to approximate things like we saw in the last paper with the adhesion contact points being these black lines. There's a whole process underlying that. But we don't care about that just to get a handle on their general functional role. But of course, you need to have those details. And if you have a better model that can incorporate those details, then that's something that you should maybe consider to be a better hypothesis. In any case, so they talk about how the machine metaphor is embedded in all of our thinking about molecular biology, and how we. Oh, go ahead.
Sorry, they're quoting Belousov, so this is good. Dick will approve.
Okay, yeah.
Yes.
Oh yeah, here he is, Belousov. And so yeah. In most discussions about laws, principles, and unifying frameworks, beginning at these relatively high levels of complexity. So again, we're talking about different laws and principles in physics, and one of the aims of physics is to extract laws from data, so that first of all, nature is lawful, and that we can kind of extract laws that we can predict and rely on. And if you're doing something like bioengineering, that's a very important thing to want to have, because you want to be able to exploit that to build controllable systems. But on the other hand, you can't just invent those laws. They have to be there, and they have to be there at a high level of complexity. Because if they're just kind of trivial laws, then it doesn't really do anything for anyone. And so this popular view of the cell as a factor for producing molecular machines says little about the underlying rules of life, although the gene is an essential source of information in the production of proteins and other molecular processes. Nicholson noted that its products are released into a cellular milieu that already possesses spatial structure, and this is shaped by pre-existing self-organizing processes. So, if you think about a cell, you have all these molecular processes going on, but there is also there's already this spatial structure that exists. Sometimes it's compartmentalization. Sometimes it's just kind of the way that things are organized within the cell, like organelles, or the process of cell division or whatever. And sometimes, of course, it's this organization between cells, where you have cells that interact. You might have like secretions that occur through molecular processes that guide collective behavior of cells. You also have a lot of complexity at the cell membrane, and that, of course, controls things like adhesion and other types of signaling through surface receptors and things like that. So these are all kind of have to be accounted for in a theory like this. There's all this existing self-organizing process that when you have a process that say evolves, or you have like a process that you're measuring in terms of its molecular content, that's organized around a lot of existing spatial structure. We have to take that into account in our models. So what are the basic principles that enable an organism to develop, self-organize, operate in the most energy efficient way, and remain intrinsically stable? And that's what we're trying to do. And we're trying to find, in particular, the super stable state where you have all of these different changes over time and it's stable nonetheless. So we want to have the super stable state. That's what we're looking for. And so, it kind of goes into the idea of laws in biology and how the laws of motion and thermodynamics are considered universal. But a lot of these complex processes within cells, we don't really understand the laws of motion and thermodynamics in the context of a lot of these biological processes at the cellular level, anyways. Like, cell function, development, reproduction, regeneration. And so, we kind of call a lot of this epigenetics and kind of throw that into that bin. So if you have interactions between cells and tissues, we have, different things that are what we might call extragenetic. And there's just this box of change called epigenetic processes. And so this is, maybe not very useful for really understanding a lot of complex behaviors and thinking about things not as kind of hard mechanical systems, but soft matter systems. And, there's a whole field, of course, of soft matter physics that people have been applying to developmental biology in the last couple decades. And, there have been a lot of interesting findings about, populations of cells in tissues, how tissue sheets behave collectively, how cells migrate collectively. But there's still this aspect of not understanding at the soft matter level what the laws are and how thermodynamics operates. So there's still this need for a unifying theory around this sort of soft matter perspective and more generally around mechanics and physics that govern these kind of systems. So that's where they introduced tensegrity. They define tensegrity as the unification of tension and integrity. So it's this portmanteau of tension and integrity, and then describes a structural system that consists of a set of discrete compression elements, which are these struts. So like in the last paper, we saw the nodes and edges. The edges here are these struts, so they're physically manifest as struts. So they actually introduce compression on these different, the struts are the nodes, and the cables are the things that are tensioned. So the discrete compression elements are struts. They're these nodes, and you can see in this model here where you have sort of the nodes and the edges, and then you have the nodes, which are the struts, and these are compression elements, and then they're suspended within a network of tensioned elements or cables. So again, you can push on a cable and it introduces tension. You can pull on it, you can compress it, and then that affects the rest of the network. And then the activity of the struts can also affect that as well. It can drive this process. It can be internal to the network or external, I guess. So this is where they kind of they review, first of all, Buckminster Fuller, who was the person who originated tensegrity actually in architecture. And you've seen the buckyball, which is the C60 element, where you have a structure of carbon, and that's a what they call a buckyball, which is a Buckminster Fuller inspired structure. He's also built, if you've ever seen a geodesic dome in the wild, those are these structures that are half circle or half spheres, and they're held stable, as a product of tensegrity. And so then this made its way into biology around the late 90s, into the aughts, and they have these citations here where there's this new structural principle where you don't necessarily have a traditional physical principle. There's this principle of kind of I guess that first of all, that you have this sort of dynamic tension, and that things are held in dynamic tension. And the other aspect is that this is self-organized in some way. So you have to have this structure, whatever it is. It's you could think of a cell as being the structure, a cell membrane, and it's held in stability. But how does it sort of form? Does it form by a single node and then another node and then connecting with um, some sort of connection and then having additional nodes added? How does that form while also retaining that same stability? So, this is kind of maybe the problem of origins for biotensegrity, where you have this complex structure you can observe, but how do you start off with that structure? How do you get it started? And then, of course, how does it get maintained over time? So, tensegrity models are strong, flexible, and resistant, and physical properties that are similar to living tissues in many ways, and they have some citations here. They change shape with the minimum of effort and automatically return to the same stable position of equilibrium, irrespective of the direction of gravity, and remain intrinsically stable. When one part changes, the entire system becomes involved, thus enabling forces, power, and information to be transferred from one region to another in a controlled way. So these are the stress strain curves for materials. And so this is A is Hooke's law, which is linearity over this region of the stress strain curve, and then so this is where you have an input of stress and an output of strain, or an input of strain and an output of stress, and that's a linear relationship under Hooke's law. Then in B and C you have the nonlinear behavior of tensegrity in living tissues. So you have these quasi-linear ranges, but they're decoupled, and the whole regime isn't linear. So you might expect for some strain values, the stress values to be linear, and then there's this phase transition where the stress values are much different given the input strain values, and then you have these quasi-linear regions which are local but not global.
Yeah, they have a change of state.
Yeah, change of state. This is an example here in C. The physiological range is this part of the quasi-linear region here, and then microfailures are in this second linear region. So you might go from like the normal physiological range to this phase transition where you have a failure or collapse of the system or some breakage, and so then you have this difference in the stress strain curve, and you have to undergo a phase transition to get there, but it's very different behavior, but it's basically the same structure. And then D through F are some further examples of complex tissue fluid behaviors, which we call soft matter behaviors. So you might have different stress strain curves for different materials. So if you are modeling a cell, collagen might have one behavior and cortical bone might have another behavior. And of course, you have to account for that in your model. So if you have a model of a limb where you have cortical bone, hard bone, and then you have collagen and you have skin and you have muscle, those all have different stress strain curves. So you have to account for those in proportion when you're modeling the actual limb of an arm or a leg or something like that. Then this is E is where you have constant stress, where you have creep and relaxation, and then stress over time, which is thixotropy and rheopsy or rheopexy, I guess. Was that?
Yeah, it's okay. The names. I'm laughing at the names.
So this is this first one. E is time versus strain. So you're looking at strain over time, and then the second one is stress, time, and viscosity. So it's like this stress over time and then viscosity. And this is kind of the behavior of the material. So thixotropy is like if you take a ketchup bottle and you kind of turn it upside down and the ketchup starts flowing. That's a thixotropic medium. has different behaviors, right?
That works with clay as well.
Okay, yeah,
it's very hard, and then you mix it up, and then it flows.
Yeah,
that sort of material. And cells are like that quite often.
Yeah.
I still would like to see a stress strain curve when I'm reading about these things, instead of just, oh, well, here's the strain time curve, like the creep or relaxation. That's fine too, actually, but I still was looking for stress strain curves, and I would often get, oh, this is a creep and relaxation behavior. Anyways.
Yeah. So, like, for example, with F, if you apply stress over a certain amount of time, the response isn't immediate. The response builds up.
Yeah.
And there's a change in viscosity as the stress accumulates over time, or rather, if you apply a stress maybe over a minute, say, rather than 5 seconds, you get a different, you get a flow versus no flow. So if I just kind of turn a ketchup bottle over briefly and then put it back right side up, it doesn't really flow. But if I turn it over and hold it upside down, it's introducing stresses and it's pushing it out and it's flowing. So it's like you have to give it like this kind of initial input, or this collective input of stress to get it to move. And so that's kind of the idea there. And, that's just a property of the material, a property of that flow.
It acts like crazy. Is it crazy putty? Or no, silly putty.
Silly
That's
putty,
it.
yeah.
Yeah, silly putty. Silly putty.
I think we need a crazy putty, actually.
Yeah. Cells are crazy putty.
Yeah. So there are two components here. One is that there's this behavior that changes over time, and that's nonlinear. And then there's this other component where you have this sort of composite nature of things. So, like, not every, if you have if you're trying to model a system, not every component of the system behaves the same way. You can't assume homogeneity. You have to assume some heterogeneity, and you have to assume some sort of nonlinearity over time.
Yeah.
And so, yeah.
Mm-hmm. Yes. And it definitely has a time component, because the faster you compress it, the stiffer it'll be. That just, and it's kind of obvious when you think of it. But different tissues have a different time components too.
Right.
As to how fast they can react.
So they talk about biotensegrity as embracing the principles of self-organization. So they show this example here where cells are coming into contact, like we saw in the last paper. They're forming these networks or clumps of cells. These networks become more complex, and then those networks form these sheets of cells that do things collectively. So you have two cells that are existing in isolation. They come together, they might form, of course, some sort of adhesion, or they might, kind of form a boundary. And then, of course, if a number of cells do that, you form this network, and then more cells join in. You get cell division, you get this more complex network, and then you get a tissue.
I would prefer to see that last diagram C as a group of bubbles.
Yeah, yeah.
Rather than that, because those are still disassociated from each other, more or less.
Right. Right. Yeah, they have the networks here, but they don't show it here, or the sort of what this was
shape.
Yeah.
Final shape. Yeah.
And then we talk about closed kinematic chains here. So this is kind of getting into this linkage system idea, where you have multiple bars, which is this linkage system, maybe from the origin of a force to its output. And these multiple bars will move kind of they're maybe hinged, and they move kind of together. They have to be coordinated by some force. They are coupled into these continuous mechanical loops, with each one influencing the other's position. So as you move outward from the source of the force, these bars will coordinate their movement. So if you think about like um, dual pendulum, if you've ever seen one of those, where you have, if you have a single pendulum, of course it moves back and forth, and it's deterministic in its movement. But if you have a double pendulum, where you have one pendulum and then a second pendulum that's hinged onto the bottom of the first one, those dynamics are much more complex because you have two pendula that are kind of moving semi-independently. There's still one influences the other, but there's this one,
Yeah.
yeah,
That's an example of a chaotic system.
right.
A
Getting
dual
into that
pendulum.
region, yeah.
Yes, and tensegrities are like that. When you have two bars attached to each other, which you would get in cells. So it tends to be a chaotic system, a constrained chaotic system, but it's still a chaotic system. It depends on initial conditions.
Right, yeah. So, I mean, that's the idea of like a kinematic chain, which we talked about before, which is this, where you have this chain of movement outward, and it gets less controlled as you move outward. But of course, in let's say like a cell, like we've been seeing, we don't have an open kinematic chain. We have a closed kinematic chain. So that means that you have like this double pendulum, but it's connected back to the origin, which is interesting.
Yeah, I can tell you all about it.
Yeah.
Like the computer program that would never work.
Yeah.
Yes.
And of course, as Susan says, it doesn't make it any easier to predict just because it's connected back to the source. Because it's usually more than like a double pendulum. It's a multi segmented pendulum, and then it's connected back to the source, and that the source has to respond to that force that's now being brought back to it. So now it has to regulate itself, and there's this.
Yes, and on top of that, these linkages change.
Right.
Because it's a living cell, of course. Yeah.
So they show their model here, a 4-bar mechanism showing the pin joints, trajectories, and limits of 3 moving bars in relation to a fixed or horizontal reference bar, and then the changing angles, alpha over beta, that define its nonlinear behavior. So this is all kind of the way this is. And of course, this has a geometry. In the cell, we're going to have a specific geometry. It's not just kind of a pendulum moving, freely in space. It's this constrained geometry, and then you have these forces operating in this system. So, this is kind of the problem here. We have a very complex system because, as we just mentioned, a double pendulum can produce chaotic behavior, and that's pretty complex. And we have this multi-jointed pendulum that's connected back to the origin, and it basically, what that means is that it has to, all of the forces in a system have to be kind of responded to by other components of the system to maintain its stability. So and then of course, if you use like a classical physics program to model that, it's going to give you a lot of failure because it's not going to understand that kind of feedback, and it's not going to understand the different aspects of that. So these closed kinematic chains have been described in the feeding mechanisms of fish, shrimps, and humans, the fascicle connective tissues, and the limbs of horses and humans. So, this is just kind of going into some of the biomechanics of kinematics, or biomechanics of tensegrity. And then, of course, in kinematics, we use algebraic equations to describe the geometry of motion. But, of course, in tensegrities, we have to think a little bit differently. We basically are mimicking the basic mechanics of tensegrity by using these closed chain kinematics, but we need maybe something more than algebraic equations to model this. So this is another tensegrity model, the T-ICOSA model. So they have, what are these called, Susan? Where you have the 3 bars and the 4 bars.
The 3 bars and the 4 bars?
Yeah, like these examples here.
Um,
I mean, what are they usual? What's the sort of the jargon that they use for it?
oh, okay. I'm drawing a blank. So,
Okay.
yeah, there's a T3 and a T4.
Oh, okay. Maybe that's all.
Yeah, so it's calling it a TI CASA model. So, yeah.
Yeah.
A T3 is a little pyramid.
Okay. Yeah. Yeah.
Kind of. Anyways, is it a pyramid? Sorry, it's 3-sided. It's a 3-sided tensegrity is called a T3.
Yeah. Yeah.
Yeah. Anyways, and it's the simplest one, and it's very stable again because it's triangles.
Yeah.
And you can do all kinds of things with T3s in an ordinary simulation package that you can't do with the T4 or any of these other shapes because they're very stable. That's why I chose T3s for my connections between cells, and well, they are a triangle anyway. Nature discovered T3s way before we did. Yeah, because that's the trijunctions.
Right.
Are essentially a T3.
So they give maybe some biological examples here. Monoarticular muscles might then be considered as a variable length set of bars within 3-bar systems, or T3s, while bioarticular isometric muscles can form tensionally stiff bars within 4-bar systems, or T4, that enable multiple joints to be coupled in ways that transfer power from one part of the body to another in a controlled way. So there are these structures called muscle synergies, which are where you have like a bunch of muscles that are activated during the movement, versus just kind of like looking at single muscles as they articulate on the joints. So you're getting this different level of coordination. You're getting the one layer of coordination with the T3 and another layer of coordination with the T4. And I don't have the cellular analogy to that. I guess, maybe something with actin molecules or something where they're acting and maybe collectively versus independently. That might be one way to think about it. But this just shows this difference between the muscle synergy and the muscle movement, and kind of how this is related to a closed chain geometry. So the muscle is of course reacting to it's being stretched, and muscle synergies are reacting to some intentional input or output, and they're coordinating accordingly. So you have this sort of, you can think about muscle movements in the body, in our bodies, as an example of this closed chain geometry.
Okay, and the bones are the rods, and they're a very important part of the system.
Right.
But what operates the system, of course, are the muscles in blue here, shown in blue.
Yeah. The muscle provides the bone provides the underlying stability, and the muscle provides the movement, the coordinated movement, where it provides maybe some power to that movement, I guess. And so, yeah, that's what it looks like. They just give that example. And then, so they kind of go through that. Then they talk about structure, stability, and energetics, where the concept of biotensegrity is predicated on anatomy as the physical representation of a hugely complex pattern of interacting force vectors. So it's kind of what we saw in the muscles, in the bone, where there's this set of interacting force vectors, and the body is sort of the structure that's organized around. And then there's a sufficient separation of these into different structural modes, cable/thread/bars, as their closed chain interactions that allow such systems to change shape while remaining intrinsically stable throughout. So I think they talk about figures 4 and 5. Well, that's way up above. But figure 7 shows this tensegrity portrayed as an energy framework. So you have in A, you have this energy framework with these red lines and these blue lines. The red lines shown in B are isolated sets of points, which are the nodes and vertices. So you have the nodes which are isolated and then they're connected by vertices. That's sort of the physical connection between them. And then C are the point combinations, which are the edges that can get closer together but not further apart. Then they say point
compression. Yeah.
Yeah. Point combinations are edges that get further apart but not closer together.
In tension.
Yeah.
Yeah.
So they show that there's this structure that has two components. You have sort of the isolated set of points, and then you have the point combinations. And that's kind of the way to model this. And then the stability of the framework is then achieved through coupled changes in the elastic strain energies, as the springs or the edges automatically seek their minimal energy states. So these edges are always trying to minimize their energy. But when you have forces maybe operating from the outside, or when those points need to make changes in their orientation or their organizational state, then you get these disruptions in the minimal energy state. So you can think about it like in homeostasis, where you have a homeostatic state, which is a relaxed state, where everything's at minimal energy. And then you introduce perturbations to that. So you stretch on something, or you push something together, or you change its orientation. Maybe the cell changes shape, or the muscles get activated, the limbs change their orientation. And those are all introducing, energetic states that are not minimal. So then the system has to find its way back to the minimal energetic state. And in doing so, it has to find its way back to equilibrium or to homeostasis. So there's some homeostatic set point that the system exists at. It's moved in different directions, and then it has to find its way back to equilibrium, back to the homeostatic state. And that's how you achieve superstability, in that you can perturb the system in a lot of ways, and you can get these nonlinear responses that occur in kind of returning back to that minimal state, that homeostatic state. And so, that does raise an interesting question, which is that you can change the number of edges and the number of nodes in the system as the anatomy evolves, or as it develops, or as you just simply get deformations in the shape. So, if you're talking about like a cell and it's dividing, that cell divides, and there's this process of kind of breaking this tensegrity structure into two parts, or replicating the tensegrity structure, but doing it in a way that breaks its continuity. How do you recover that shape? How do you recover that structure? And so this is kind of what they're getting at in this section. They talk about how the edges can be freely split into shorter ones with new vertices, thereby increasing their total count, or connected into longer ones that decrease the number of vertices, all without compromising the system stability. So the edges can get longer, shorter, divide, or merge together, or even switch energy functions. And the configuration as a whole remains completely stable, no matter how complicated it becomes. And I guess that's true. I mean, I'd like to see like some simulations where you really try to break the tensegrity structure. Because at some point there must be a failure mode, and it'd be interesting to know what that is. But that does speak to, like, if you had something like cell division, where you could change the network, you stretch it, sometimes you break its continuity, but it can recover from that.
Yeah.
Yeah.
Yeah, breaking is good sometimes.
Yeah.
Especially if you're cell dividing, or even muscles. You stretch them. That's how you build more muscle mass, as you overstretch them.
Yeah.
So, yeah.
You get disintegration of the existing muscle mass, and then it rebuilds. You get, yeah.
Yeah, you overstrain it, and you can build more muscle.
Yeah. So, in summary, an appreciation of living organisms as complex, constantly changing, multi-scale energy frameworks that continuously flow from one stable state to another, maintain overall viability through their myriad homeostatic processes is what we're talking about here. So this is really a lot. They kind of lay out their theory here and how this works. We just have this minimal energy principle. So we actually do have, you don't want to call it the free energy principle, you can, but it's very similar to that idea. And so it's really interesting. We're dealing with a complex system. We're dealing with something that's self-regulating. We're dealing with something that's self-organizing. We're dealing with something that exists at multiple scales of organization. And then we have this sort of self-repair aspect where it can remodel and repair itself and with minimal effort. So then in the discussion, they talk about how there are these differences between structural and functional theories. And then kind of, structuralism is kind of separated from functionalism in ways. And, we ended up with, these two distinctions between structural theories and functional theories of biology. And so now kind of they're bringing those together, and that's really what we need. And they talk about the laws of biology as being also this thing needs to be kind of reconsidered in light of things like tensegrities. And they bring up this point: stability before mobility. So in order, we think about like cell motility, say, as like a defining transition, evolutionary transition. When things started to move around, that's when you got a lot of interesting things in the history of life. So when you could have like a predator chase after a prey, or a prey evade a predator, when you had cell migrations, or when you had things moving and changing shape, that's when things got really interesting. And so this is talking about how things are unstable. So the structure has to come before mobility. So you can't have mobility without structural stability. And so orthodox biomechanics has tended to focus on mobility with stability as a secondary consideration. But it's the other way around, where you need to have stability first and then mobility, and mobility has to catch up to stability rather than the other way around. Otherwise, you have this intrinsically unstable system. So this is where, they talk about sort of the movement of muscles, and they talk about how, what comes first, the muscles or the neural system. Sometimes you need to have the neural system in order to sort of drive the muscles. But yeah, we know that muscles can twitch on their own and have behaviors on their own. And we also know that there's passive dynamic walking, which you can achieve just purely by mechanical means. So there's probably this aspect of muscle stability that exists independently of the nervous system. Then the nervous system provides an input, which provides the input of enhanced mobility. But you need the stability first in order to get to that point. And then they kind of use a lot of examples from the human musculoskeletal system. And what we're kind of interested in, of course, is cells, but also maybe like other types of musculoskeletal systems. So it's interesting that they kind of use this analogy because we're, of course, actin is our analogy in the cell, but also that we can think more broadly about these closed chain systems, these closed chain kinematic systems, and the homeostasis that drives superstability. So I think that's enough for that paper, but I think it's an interesting theoretical foray into this. They bring up a number of concepts that are really, really useful. No modeling, though. No modeling.
Well, yeah, I would like to see them try to model that.
Yeah.
They're probably trying.
Oh, yeah, I'm sure. It's hard to do, I guess. Numeric. Yeah. Okay, so that's it for today. Thank you for attending, and see you next week.